In this chapter, we are going to look at the smell and the taste, as well as part of the ear and its function. So, smell, olfaction, and taste, gustation, is a it's complementary senses which let us know whether a substance should be savored or avoided. Chemoreceptors are used by these systems, and with related to the chemoreceptors, the chemicals must be dissolved in the aqueous solution to be picked up by them. Smell receptors are excited by chemicals which is dissolved in the nasal fluid, and the taste receptors, they respond to the chemicals dissolved in saliva. So let's first look at the smell. So location and the structure of the olfactory receptors. Olfactory epithelium is the organ of smell, and it's located in the roof of the nasal cavity. It covers the superior nasal concha, and it contains the olfactory sensory neurons, which are bipolar neurons with radiating olfactory cilia. The supporting cells surround and cushion the olfactory receptor cells. The olfactory stem cells, they lie at the base of the epithelium. So here you would see the olfactory nerve endings, which they lie at the roof of the nasal cavity. You see the olfactory epithelium lining this area at the roof of the nasal cavity. The olfactory neurons are bipolar neurons, which is an important factor. The thin apical dendrites of them terminates in the knob, and also these bipolar neurons are long, largely non-motile cilia, and we call them as olfactory cilia, and these olfactory cilia radiate from the knob. And it's been covered by the mucus, which is the solvent for the odorants. Bundles of non-myelinated axons of the olfactory receptor cells gather in fascicles, which make up the filaments of olfactory nerve, which is the first cranial nerve. And olfactory neurons, unlike the other neurons, they have stem cells, which give rise to new neurons every 30 to 60 days. So if even the epithelium is sheds off, and we know the fact that the neurons, they are non-dividing, but the stem cells, they can give rise to new neurons. So here you would see the olfactory cilia, the dendrites of the bipolar neurons here, and the olfactory bulb, and they synapses with the mitral cells, and then at the end all the axons, which creates the olfactory tract. So let's look at the specificity of olfactory receptors. Smell would contain hundreds of different odorants, and humans have approximately smell genes which is active in nose. And these genes encodes a unique receptor protein, the protein which responds to one or more odors. Each odor can bind to several different receptors, and each receptor has one type of receptor protein. In addition, the pain and temperature receptors are also in nasal cavity. They respond to irritants such as ammonia, or can smell hot or cold, such as chili peppers, which is hot, and menthol, like for cold. In order to smell substances, the substance must be volatile. It must be in gaseous state. So odorants must also be able to dissolve in olfactory epithelium fluid in order to smell that. So activation of olfactory sensory neurons happens when the dissolved odorants bind to the receptor proteins in the olfactory cilium membranes. And once they bind into it, they open cation channels, generating the receptor potential. And at threshold, the action potential is conducted to first relay station in the olfactory bulb. The odorants bind to receptors, activate the G protein, which is referred to as the G-olf. The G protein activates, causes cyclic AMP, which is the second messenger, and this cyclic AMP opens up sodium and calcium channels, and the sodium influx causes depolarization, and then impulse transmission happens. The calcium influx causes decreased response to the sustained stimuli, refers to as the olfactory adaptation. So, this you might have experienced, like people can't smell a certain odor after being exposed to it for a while, or you get adapted. For instance, if you work in a hospital, like sometimes the detergent and so on, you would smell it, and after a while your nose get adapted to it, so that's why it's called the olfactory adaptation. Here it also shows in a figure the activation of the olfactory receptors by its chemicals and generating the second messengers, opening up the sodium and the calcium channels. Let's look at the olfactory pathway now. The filaments of the olfactory nerves, they synapse with the mitral cells located in the overlying olfactory bulb. The mitral cells, they are the second-order neurons that form the olfactory tract. The synapse occurs in structures called glomeruli, and the axons from neurons with same receptor type, they converge on a given type of the glomerulus. Glomeruli is the plural, glomerulus is the singular word. The mitral cells, they amplify, they refine, and they relay signals. So there's granules called amacrine granule cells. Amacrine granule, which is in amacrine granule cells, they release a neurotransmitter called GABA to inhibit mitral cells so that only highly excitatory impulses is going to be transmitted through the mitral cells. Impulse from activated mitral cells, they travel via olfactory tracts to the piriform lobe of the olfactory cortex of the brain, and some information sent to the frontal lobe, and some passes through thalamus first. So when it's passed through the thalamus, the smell is consciously interpreted and then identified. Some information then sent to the hypothalamus, amygdala, and other regions of the limbic system as well. So if they sent this information, then the emotional responses to odor is going to be elicited. The clinical scenarios involved with olfaction would be the anosmias, which is an olfactory disorder most resulting from head injuries which tear the olfactory nerves. And after effects of it could happen due to the after effects of the nasal cavity inflammation, or certain neurological disorders such as the Parkinson's disease. And also this condition called olfactory hallucinations, which is usually caused by the temporal lobe epilepsy that involves the olfactory cortex. And in this case, some people have olfactory auras prior to the epileptic seizure that they would complain about, that they smell some weird smells, and then they have this epileptic attack. Let's look at the sense of taste, and first the location and the structure of the taste buds. So now we are moving to the taste and its structure and the physiology. So taste buds, it's the sensory organs for taste, and most of 10,000 taste buds, they are located on tongue in papillae, which are peg-like projections of the tongue mucosa. And there are several types: fungiform papillae, foliate papillae, valate papillae. The fungiform papillae, tops of these mushroom-shaped structures, house most taste bud, and it's scattered across the tongue. The foliate papillae, they are on side walls of the tongue. The velate papillae is the largest taste buds with 8 to 12 forming a V shaped at the back of the tongue. And they're arranged in kind of a V shaped. And there are few on the soft palate, cheeks, pharynx, and the epiglottis. So here you would see the foliate papillae, which is at the lateral side, the fungiform papillae, which is all around the tongue mostly, as well as the velate papillae over here. Each taste bud consists of 50 to 100 flask-shaped epithelial cells of two types. And the gustatory epithelial cells, which are the taste receptor cells, have microvilli called gustatory hairs, and that projects into taste pores, which is bathed in saliva. And the sensory dendrites coiled around gustatory epithelial cells send taste signals to brain. There are three types of gustatory epithelial cells. One releases serotonin, others lack synaptic vesicles, but one releases ATP as neurotransmitter. The basal epithelial cells, they're dynamic stem cells that divide every 7 to 10 days. So here it would, they have shown here the velate papillae, as well as the enlarged view of the taste bud, which is the gustatory epithelial cells, and the gustatory hair, and so on. There are 5 basic taste sensations: sweet, sour, salty, bitter, and umami. So sweet is for sugar, saccharine, alcohol, and some amino acids, and some lead salts leads to the sweet sensation. The sour is from the hydrogen ions in the solution, and salty is from the metal ions, inorganic salts, and sodium chloride's taste is the saltiest. The bitter is due to alkaloids such as quinine and nicotine, caffeine, and non-alkaloids such as aspirin. The umami, the amino acid glutamate and aspartate, example like in certain meat, beef, or cheese taste, and the monosodium glutamate is monosodium glutamate give rise to this taste sensation. It says like this could be a possible 6th taste growing evidence. Humans can taste long chain fatty acids from lipids. Maybe it's think like that's why people would like for fatty foods. And taste likes or dislikes have homeostatic value, and it leads to guide intake of beneficial and potentially harmful substances. And if your dislike for sourness and bitterness is a protective way of warning us if something is spoiled or poisonous. So let's look at the physiology of this taste. For someone to able to taste a chemical, there are 3 things needs to happen. It should be dissolved in saliva, diffused into taste pore, and contact gustatory hairs. So binding of food chemical, or we call it as taste, and depolarizes the cell membrane of the gustatory epithelial cell membrane, and it causes release of neurotransmitter. And this neurotransmitter then binds to the dendrite of the sensory neuron and then initiates a generator potential, which leads to an action potential. Different gustatory cells have different thresholds for activation, and it's been said that the beta receptors are most sensitive. All taste receptors, they adapt in 3 to 5 seconds, with complete adaptation happens in 1 to 5 minutes. And then the gustatory epithelial cell depolarization is happening, which is caused by salty taste is due to the sodium ion influx, which directly causes depolarization. Sour taste is due to hydrogen ion acting intercellularly by opening channels which allow other cations to enter. And some unique receptors for sweet, bitter, and umami, but all are coupled to G protein gustudecine. So the gustudecine activation causes release of stored calcium ion that opens cation channels, which causes depolarization and release of neurotransmitter ATP. Related to the pathway of gustatory sensation, there are two main cranial nerve pairs carry taste impulses from tongue to brain. That is, the facial nerve and the glossopharyngeal nerve. The facial nerve, it carries impulses from anterior 2/3 of the tongue, and the glossopharyngeal carries impulses from posterior 1/3 and the pharynx. The vagus nerve is also involved in transmit from epiglottis and the lower pharynx. What will happen is the fibers, they synapse in the solitary nucleus of the medulla and then travel to the thalamus and then to the gustatory cortex in insula. So the hypothalamus and the limbic system is involved, and it allows us to determine the appreciation of taste. And here it would show us the pathway, the glossopharyngeal nerve from the posterior 1/3, the facial nerve from anterior 2/3. They rely at the solitary nucleus of the medulla and then go across the thalamus and to the gustatory cortex in the insular area. The important roles of taste involves triggering reflexes involved in digestion, such as increasing secretion of saliva into the mouth when the food is present, increased secretion of gastric juice into the stomach when even the food is in the mouth. It may initiate protective reactions such as gagging or reflexive vomiting when you encounter new food that your body is not used to or such like that. Taste is 80% smell, and if nose is blocked, you might have experienced that foods taste bland. Mouth also contains thermoreceptors, mechanoreceptors, and nociceptors. So temperature and texture enhances or detract from taste. Spicy hot foods, they can excite pain receptors in the mouth, which some people experience as pleasure. So that's why some people would like to eat more spicy. Some people would experience it as so pain. So some of the clinical importance is that taste disorders are less common than disorders of smell, mostly because taste receptors are served by 3 different nerves, and not likely that all 3 nerves would be damaged at the same time. And also causes of taste disorders could involve the upper respiratory tract infection, head injuries, chemicals, medication, head and neck radiation for cancer treatments, such like that would lead to taste disorders. Okay. Yes. So now let's go over the hearing and balance. First, look at the structure of the ear. The ear has 3 major areas: the external ear for hearing only, the middle ear, that's hearing only, and the internal ear, which has an additional function in addition to the hearing, is that the equilibrium. Receptors for hearing and balance respond to separate stimuli, and they are activated independently. So let's go over the external ear first. The external, or we call it as the outer ear, consists of 2 parts: the auricle, the pinna in lay language, or external acoustic meatus, we call it as the auditory canal. The auricle is shell-shaped structure surrounding ear canal that functions to funnel sound waves into the auditory canal. The cartilaginous rim is called as the helix. The fleshy earlobe is called as the lobule. The external acoustic meatus is a short curved tube lined with skin-bearing hairs, sebaceous glands, and ceruminous earwax glands. So it transmits sound waves to eardrum. The tympanic membrane is the eardrum and its boundary between the external and the middle ears. The thin translucent connective tissue, it's a thin translucent connective tissue membrane. It vibrates in response to sound, and it transfers sound energy to bones of the middle ear. Here you would see the helix and the lobule, the fleshy part, the cartilaginous part, the pinna, the external acoustic meatus, and the tympanic membrane. The middle ear, the tympanic cavity, is a small air-filled mucosa-lined cavity in the temporal bone, and it's flanked laterally by eardrum and medially by the bony wall containing oval and round membranous windows. The epitympanic recess is the superior portion of the middle ear, which is the roof of the cavity. And the mastoid antrum is the canal for communication with the mastoid air cells in the mastoid process. So here it shows the entrance to the mastoid, which is the antrum in the epitympanic recess. And this part is included in the middle ear. This is the auditory tube, the pharyngotympanic tube. The pharyngotympanic tube connects middle ear to the nasopharynx, and it's formally called as the Eustachian tube when you look at the older textbooks. It's usually flattened tube, but can be opened by yawning or swallowing to equalize pressure in the middle ear cavity with the external air pressures. The tympanic membrane cannot vibrate efficiently if the pressure on the both sides are not equal, so the sound can be distorted in that case. In the middle ear, there are auditory ossicles, and there are three small bones in the tympanic cavity named for their shape. The malleus, which is the hammer, is secured to eardrum. The incus, anvil, the stapes, the stirrup base fits into the oval window. You can remember this as MIS, as in order, M-I-S. The synovial joints allow the malleus to articulate with the incus, which articulates with the stapes accordingly. And suspended by these three bony ossicles, they're suspended by ligaments. These transmit vibratory motion of the eardrum to the oval window. The tensor tympani and the stapedius muscles, they contract reflexively in response to loud sounds to prevent damage to the hearing receptors. And here it shows the malleus, the incus, and the stapes. Clinically, this is a common problem, the otitis media. It's a middle ear inflammation, commonly seen in children with sore throat. And because it's especially those with shorter, more horizontal pharyngotympanic tubes, because it could get obstructed and then that could lead to inflammation as well. Most frequent cause of hearing loss in children is the otitis media. And what will happen is the acute infectious forms cause eardrum to bulge outward and become inflamed. And most cases, it responds to antibiotics. The internal ear is also referred to as the labyrinth or the maze, and it's located in the temporal bone behind the eye socket. There are two major divisions, the bony labyrinth and the membranous labyrinth. The bony labyrinth is the system of tortuous canals and cavities that worm through the bone. It divided into three regions, the vestibule, semicircular canals, and the cochlea. And It's filled with perilimb fluid, which is similar to the cerebrospinal fluid. The membranous labyrinth is a series of membranous sacs and ducts contained in bony labyrinth filled with potassium-rich endolymph. And here you would see the semicircular ducts in the semicircular canals, the vestibule area over here, and then the cochlea, which is curled up. The vestibule is the central egg-shaped cavity of the bony labyrinth. It contains two membranous sacs, the saccule, which continuous with the cochlear duct, and the utricle, which is continuous with the semicircular canals. And the sacs, it house equilibrium receptors regions that responds to the gravity and changes in the position of the head. The semicircular canals, they are three and oriented in three planes of space: anterior, lateral, and posterior. The anterior and posterior are at right angles to each other, where the lateral canal is almost horizontal. The membranous semicircular ducts, they line each canal and communicate with the utricle. And the structure, the ampulla, is an enlarged area of the ducts of each canal which house equilibrium receptor regions called the crista ampullaris. And the receptors of these crista ampullaris, they respond to angular, which is the rotational movements of the head. The cochlea is a small spiral conical bony chamber size of a split pea, and it extends from the vestibule, coils around bony pillar, we call it modulus, and contains the cochlear duct which houses the spiral organ and ends at the cochlear apex. The cavity of the cochlea divided into three chambers: the scala vestibule, the scala media, and the scala tympani. The scala tympani and the vestibule are continuous with each other at the helicotrema, which is at the apex. And the vestibular membrane, it's the roof of the cochlear duct which separates the scala media from the scala vestibule. The floor of the cochlear duct composed of bony spiral laminae and the basal laminae, and that supports the spiral organ. And the spiral organ contains the cochlear hair cells functionally arranged in one row of inner hair cells and three rows of outer hair cells. And these hair cells are sandwiched between the tectorial and the basal basilar membranes. The cochlear branch of the nerve 8, cranial nerve 8, runs from the spiral organ to the brain. So here you would see the cochlear nerve division of the vestibular cochlear nerve, which innervates the cochlear duct. Here again, it shows the scala tympani, which contains this perilimb, as well as the spiral ganglion and the spiral organ here with the basilar membrane, which lies here. And you would see the nerve is sandwiched between the spiral organ and the basilar membrane. Again, it shows a higher magnification of the description above. So this is the end of the presentation C. And as I mentioned before, you would find more information of the smell, taste, and the hearing, the structures, as well as the function in your chapter where you need to read to get more comprehension.